Good afternoon, everyone. I'm Nathalie Piperidis, and I'm very delighted to extend my warm welcome to all of you joining us today for this very anticipated webinar. I would like to begin by acknowledging the Aboriginal and Torres Strait Islander people of this nation. We acknowledge the traditional custodian of the lands on which SRA is located and where we conduct our business. We pay our respect to ancestors and elders, past and present. So today's presentation will run for about 45 minutes, followed by a traditional Q&A session. So I would like to introduce our speaker, Professor Lee Hickey, although I don't think he really needs an introduction, but it's tradition, so I'll go with it. Lee is a renowned plant geneticist and a crop breeder at QAFFI, the Queensland Alliances for Agriculture and Food Innovation at the University of Queensland. He leads innovative research and in speed breeding, in genomic selection, and in genome editing to fast-track the development of high-yielding and climate-resilient crops such as wheat, barley, chickpea, and recently is testing the world of sugarcane as well. His innovations have been adopted globally, transforming breeding programs and advancing sustainable agriculture. Lee is a prolific science communicator with more than 90 publications, including articles in high-profile journals such as Nature Plants, Nature Protocols, and Nature Biotechnology. And his research outcomes have appeared in mainstream media such as BBC, World News, National Geographic, and the New York Times, and now the SRA Webinar series. Last year, Lee also became the director of the ARC Training Center in Predictive Breeding. Lee is a passionate mentor, inspiring the next generation of agricultural researchers. He is so passionate about it that he's currently mentoring 18 PhD students. Lee, this is like 2 teams of baseball. While 20 of his previous graduates now work for leading plant breeding companies and high-profile research institutes around the globe. So to finish, I would like to mention a few of the awards that Lee has received, and I'm just going back to 2017 because instead there will be too many, I'm sure. So in 2017, he received the Australian Research Council DECRA as a Discovery Early Career Researcher Award. The same year, he received the Queensland Young Tall Poppy Scientist of the Year. And in 2019, the UQ Foundation Research Excellence Award. Then the ICM Agri-Food Award. And finally, last month, or in the last 2 months, the UQ Award for Excellence in Promoting Industry Engagement in Graduate Research. So there is like a lot more to say about Lee, but I'm sure you'd rather hear from him than from me, and all his fantastic work. So without further delay, Lee, the floor is yours.
Thanks a lot, Nat, for the introduction. I'll just pull up my presentation. It's great to be here and giving a seminar as part of the series. Okay, I hope you can all see my screen, and we'll kick things off. Thank you everyone for joining online across Queensland, Australia, and overseas as well. I hope my talk today can give you a little taste for some of the research that we're doing in this space. And hopefully trigger some new ideas for you to try in your own research or breeding programs as well. So I'd like to start with a bit of an advertisement for plant breeding. And plant breeding works remarkably well. We're very lucky we have it. In fact, it's the reason why we have very high yielding crop varieties around the world to sustain so many people on this planet. And so you can see here the trajectory of improvement of a crop. This can be just any crop. It's pretty much the same for most crops in terms of genetic improvement over time, in terms of productivity, iteratively improving the genome of that crop through plant breeding, which is fantastic. If we were to stop plant breeding tomorrow, this is what would happen. Our varieties would actually start to reduce in terms of productivity. fall victim to climate change and rapidly evolving pests and diseases that are overcoming resistances that our plant breeders are deploying. So we go backwards. Essentially, where we need to be is up here. We need to actually increase the productivity of our crops around the world. And we see breeding technologies to be a key part of this to help accelerate genetic gain, as well as bringing those improved genetics together with management practices on farm to realize those benefits. So yeah, a big challenge ahead to accelerate the genetic gain. Essentially doubling the rate of genetic gain is pretty much a goal globally. To help achieve this goal of accelerating genetic gain, we see tapping into data and converting that data into better breeding decisions to improve plant breeding efficiency is a key goal. And so this is where predictive breeding can be quite powerful and useful. Breeding programs around the world now are collecting millions and sometimes billions, depending on the scale of the breeding operation, per year. So we're collecting big data. It depends what you call a data point: genomic data, phenomic data, environmental data, you name it. But it's an opportunity really to be turning that data into better decisions. Yeah, that's where I think AI or machine learning can be quite useful for many points in the breeding process. And so this morning when I was chatting with ChatGPT, and I asked ChatGPT, well, show me the face of a future breeder using AI or machine learning. And this is what it showed me. Just kidding. It's definitely not as scary as that. It's applied statistics we're talking about, right? And so I'd like to use this figure as a bit of a framework for my presentation today to highlight some of the key areas where we see opportunities to apply machine learning or AI in the breeding process. Now, the first one, which is an area where we're not doing a lot of work at the moment, but I know large commercial breeding programs are doing this, and it's around setting the breeding goal. And you think this would be quite straightforward to determine which traits to breed for. Essentially, what does the product profile look like for these crops? But it's becoming more and more complex. And I guess there's an opportunity to tap into more data from farmers and consumers for some of our crops that could help formulate the traits that we need to package. And so I guess that's an opportunity. The first example where I'll talk to today is around phenotyping for target traits. And so let's kick off with this one. So today, UAVs fitted with sensors or even satellites are becoming a really powerful tool for phenotyping at scale that's required for plant breeding. You can see there a nice visualization of a satellite flying overhead across crops. In our research, we are using a lot of UAVs fitted with cameras or sensors to capture traits that might be correlated or used to predict traits of interest to plant breeders. Now, when it comes to this, UAVs fitted with these sensors can capture wavelengths of all different kinds of bands. Here you can see a multi-spec sensor on your screen and a typical drone that we use for our research. Now, this sort of equipment, it enables phenotyping large populations, so at scale, but it also enables measuring traditional traits more accurately, traits where the human error can come in through human assessment, because images don't lie and are very precise. But also, this also has the advantage of measuring traits that the human eye can't see. And so here you can see I've switched from RGB of this field trial to thermal, and you can see straight away we've revealed information that the human eye could not see, and that strip down the bottom there in purple is actually an irrigated strip in this barley trial, and so the plants have access to more soil water, and so they're transpiring more, and so they have a cooler canopy. This is also an advantage of using this type of technology. Now, we know that many vegetation indices that can be calculated from this imagery or canopy temperature can provide water use proxies. In fact, there's many research studies that have been done in recent years that show this really nicely. There's a correlation between these indices or even canopy temperature and water use traits. In fact, some of these researchers have also looked at the correlation between these factors and how much water the crop might have access to. And so to take this research to the next level, well, we were thinking, well, maybe we can actually use these canopy imagery to actually indirectly phenotype or predict root traits in the field. And if so, can we do this at scale, at a population scale? And if we can do that, then we can hopefully try to link this information up with our genomics and try to get to the underlying genetic controls. So that's the idea. In this case study, this research was done in collaboration with Intergrain, one of our, actually the #1 barley breeding company here in Australia. And for this study, we're working on a panel of about 500 spring barley genotypes. And you can see there on the right, the diversity in the population, really representing the diversity in modern Australian barley breeding germplasm. And this material was genotyped with a SNP chip. So we had two main experiments in this study. So one in 2022 on your left and one in 2024. And within each trial, we have different phenotyping efforts or trials. And so here on your screen, you can see in orange is the root coring trial, where we actually physically dug up the roots to measure them physically. And we use that, if you like, as a training data set to build a machine learning predictive model to predict roots on the larger main trial in blue. Now, the main trial has the large entire population, you can see. So we actually dug up the roots for only a subset of 20 genotypes that were replicated 6 times in both seasons. And that really pushed us to the limits, because we actually needed, I think, a team of 5 or 6 people for 2 weeks to dig up just 20 genotypes. So that's the real bottleneck here that we're talking about in terms of phenotyping. We couldn't even imagine phenotyping several hundreds of genotypes for this type of trait. In pink, you can see the biomass trial where we actually physically cut biomass to measure biomass at key growth stages, and we used a similar machine learning approach to predict biomass on the main trial. So we were quite fortunate in that these two environments were quite contrasting, which really set us up nicely to look at these traits in different conditions. So in 2022, it was a wet environment, much more rainfall, cooler temperatures, particularly during the flowering and grain filling period. And here you can see on your screen, less water available in the upper soil layers, particularly during that grain filling period. And this was all quantified using a crop growth model in AppSim, thanks to Corinne Chanu and our PhD student, Beta. In terms of canopy phenotyping, like I said, we measured canopy biomass, and at these key growth stages, we were able to build a predictive model doing that. So the entire trial was flown with our UAV, fitted with our sensor, a multi-spec sensor, almost weekly. And in the 2022 trial. However, less flights were conducted in 2024 at the key growth stages over the growth cycle. And in fact, that actually didn't really affect our ability to predict canopy biomass and the root traits, as you'll see later on. So digging up the roots, this is the hard work. And this is something we just never want to do again. It's a lot of work. So we worked closely with Dr. Anton Wasson from CSIRO, who has developed this really nice method called the core break method. So as the core comes out of each plot, we take 4 cores per plot, 2 intra and 2 inter-row. They get broken into 10-centimeter intervals. And then the team counts the number of roots that they can see on each break. And this provides a really nice data set for us to model down the soil profile. In the first season, we washed out some of the soil samples to validate again the relationship between root count data and root biomass. And there you can see there's not 100% correlation because of root thickness across the different levels of the soil. Now, when we visualize the root distributions down the soil profile, this is a nice way to do it. Here you can see the going from 0 to 170 centimeters. We adopted a longitudinal modeling approach, and we actually pulled out area underneath these curves, which represent different soil horizons for the soil that we're working on. And that's because the soil properties that affect the root proliferation in those zones. So we ended up with about 5 different traits, including area under the entire curve, which I guess is a proxy for root system size. And so this is a nice way to visualize the 20 genotypes that we dug up, and we modeled the root distributions in both the wet and the dry season. And straight away, down the soil profile, you can see some varieties were quite similar across the different seasons, like Rosalind, for example. But we also have varieties that behave very differently in those different seasons. And so you can see there Fathom producing a lot more roots at depth in the dry season, which might be a trait that could be useful for accessing more soil water. So, of course, we use the canopy data as well to make predictions for canopy biomass, collecting canopy data at these key growth stages. And that was the first step, was to use the raw biomass measurements at those growth stages to try to predict biomass on the entire breeding panel in that trial, where we didn't have those physical measurements. And so we used a couple of machine learning approaches. And so here you can see the results for random forest approach and a partial least squares approach. We ended up going with a partial least squares approach, but there wasn't much difference, to be honest, between the two. And here you can see now we've run that model across the entire field trial. We've scaled the phenotyping to have predicted shoot biomass or canopy biomass for the entire panel at early tillering, stem elongation, and flowering. We repeated this process for the 2024 trial as well. And then the next step was, of course, to then do the exciting part, try to predict roots using the same set of vegetation indices extracted from our drone. And so here you can see the results for 2022 and 2024. And if we look at the test set, we're getting R-squared values of between up to 0.4 to 0.6, which is I thought was really impressive for a trait that we could not see and was going down below the ground up to 170 centimeters. And so these predicted root traits actually had quite high heritability. You can see on your screen, capturing a high degree of genetic variance, which is certainly something we want to see if we want to start to select for these traits or even link to the genetics. Okay, and so once we had these predicted root traits, we then used our haplotype-based mapping approach. So if you're interested in reading more about this, we actually just put online a preprint on bioRxiv that steps through the methodology and actually compares it to GWAS outputs, which is really interesting. So it's called the local GBV approach. So we're actually calculating not a global genomic estimated breeding value where all the SNPs across the genome are summed. We're only summing the SNP effects within a haploblock to give us a haplotype effect. And now, so the first step is to estimate those SNP effects using your favorite genomic prediction model, define the blocks. We use LD, or linkage disequilibrium. In most cases, the thresholds vary depending on the population. And we then can sum up those SNP effects to give us our haplotype effects. And one of the attributes we can look at is actually the block variance, which is the variance among the haplotype effects at a single block to tell us which blocks are important for our trait. And we can visualize them in sort of a Manhattan format like this. So here's some of the results from our work on this case study, and you can see on your screen, I've shown the root-only haploblocks in brown. In green, you can see the shoot haploblocks in 2022 versus 2024, presented on the 7 chromosomes of barley. We can highlight the haploblocks associated with both root and shoot traits. So, and that's not surprising because the root and shoot are connected, and we know there's a lot of developmental genes that will modify below and above ground development. But the interesting thing for us was identifying blocks that appeared to be quite strongly associated with root traits, these predicted root traits, and not associated with shoot traits, and vice versa. So I think this really opens the door for some targeted breeding to combine these traits. For example, maintaining the same flowering window but modifying root distribution could be a goal. So yeah, a really nice example, I think, of using machine learning as a bridge to scale our phenotyping efforts on really difficult to measure traits. Okay, so back to this figure, and the next example I wanted to talk to was prediction. So predicting genotypes at the line level, and maybe machine learning approaches could be an alternative to our traditional genomic selection approach. And so when it comes to the traditional approach, BLUP is certainly that. It's been very effective. It assumes many mutations of small effect based on this infinitesimal model. It's grounded in what we know in terms of genetics. There are also Bayesian approaches to genomic prediction, and they have different assumptions. And so we thought, well, maybe machine learning approaches could present an alternative or an opportunity. And when it came to selecting the case study for this, well, I thought sugarcane was a pretty good case study because it's a complex genome, and some of the prior work done has really nicely demonstrated that that many of the target traits actually have quite a high proportion of non-additive genetic variation. So we thought maybe machine learning approaches could be useful here. So the 3 target traits in this study, in collaboration with Sugar Research Australia, were tons of cane per hectare, commercial cane sugar, and fiber content. And so the great thing about working with the breeding program is that you can get access to some beautiful data sets. And so working with the SRA team, we had in this data set, we had about 4,000 clones, genotyped with a SNP chip, evaluated across multiple regions and years. And the prediction accuracy we're comparing is the correlation between the prediction and actual performance. And so we tested 2 machine learning approaches in addition to the traditional approaches. So the convolutional neural network approach and a random forest approach. So Chen Song ran these models. They're more described in detail in his paper in the plant genome if you're interested in having a look at how he adapted these models to the data set. So I was just going to start introducing the third example or application around the crossing phase. And so we know the crossing phase is really a critical phase for any breeding program. Traditionally, that's done in terms of selection of parents is done through truncation selection. So truncation selection based on either performance or genetic merit using GBBs if you're using genomic selection. And so here on this screen, you can see if you do that, you select the best lines as parents for the next cycle. Of course, you're going to select line 1 and 2 over 3 and 4. And this is fantastic for driving genetic gain, fixing desirable alleles really quickly. However, it does come with a trade-off on genetic diversity. And so often we see lines that are discarded, like this line, for example, might have lower performance or lower GBV, but hidden in its genome are useful alleles for, say, disease resistance that are discarded during the breeding process. So this got Ben Hayes and myself really thinking around some tools that we could develop that could help with this challenge. And so this brings me to the FastStack methodology that we've been developing here at UQ over the past few years. Some of the key papers are listed below if you're interested in reading more around this. The first step really involves identifying and estimating our segment effects. And you've got to think about this as more a breeding at a haploblock or haplotype level rather than a line level. And so the first step is using our local GBV framework, which I outlined earlier, to estimate our haplotype effects across the genome. The second step is then to run our genetic algorithm to help us pick out the best parents that maximize desirable haplotypes across the genome. Now, you can work at a whole genome scale for this, which is a huge numbers problem, and that's why AI is very useful. But you can even work at a smaller scale, for example, 20 target haplotypes, where it's still challenging to optimize parent selection and crossing paths. And then once we identify the best set of parents, we then simulate all possible cross combinations to work out what's an optimal crossing path to bring those haplotypes together. And then the third step is actually doing this in practice, not just on the computer, doing it in practice, creating those real new populations. And we use speed breeding to do this really quickly. And speed breeding has been developed for many crops. I think I've been involved in developing protocols for about more than 40 different species. Sugarcane certainly is a challenge when it comes to speed breeding, being a very big plant. But I know the breeders have some tricks to induce flowering, which can be useful. So just to visualize what we're trying to do using this toolkit and using the search algorithm to pick out the best parents, we might have a set of selection candidates. It could be 20,000 breeding lines or materials. We run our search algorithm to identify which are the best parents that maximize the haplotype, the superior haplotypes across the genome. In silico, we can actually design the ultimate genotype. And this is a really good target for our algorithms to be pointing towards. In practice, the ultimate genotype is changing and shifting in time. It's also different for different regions or targets. And so that's something to keep in mind. But ultimately, what we're trying to do is assemble this ultimate genotype as quickly and as efficiently as possible. And so if you're interested in reading more about this concept on ultimate genotype, you can have a look at our latest perspective published last year. And so you'll see a common theme of some of these papers, in fact, is the technologies that we're talking about here really go across species. So it's crops and livestock. And so a common theme also for these technologies is it's species agnostic. And so not just AI is helping us to assemble these ultimate genotypes, but it's also a call to action, if you like, for some of the biotechnologies that could be useful to recombine target regions of the genome. So targeted recombination or synthetic biology approaches that could be very useful if we can develop the technology to a level where it can be applied at scale in plant breeding. So to show these tools in action, I picked out this case study. We're actually applying these tools across many crops now, including some horticultural crops, which is really exciting. But this case study is on mungbean. It's an ACR project, so it's aid funding from the federal government, and it's really, this project is aiming to develop tools and germplasm for mungbean breeders around the world in Asia and Africa. And so you can see there, spinning on your screen is the population we studied for this case study. It's a mungbean mini-core collection, so really diverse materials, almost 300 mungbean accessions, genotyped with DART-seq SNP markers. And so this is the phenotypic data we used in this study, a focus on seed weight, which is a really important trait. Some markets like really big seeds for sprouting and that sort of thing. Others actually prefer small seed size. So it's a really interesting trait to work with, very important. And thanks to the collaborators in this project, all the breeders, they phenotyped this collection at all their different locations, and we had this beautiful data set to work with. You can see there the heritability for seed weight, moderate to high, which was really nice to see. We used a FA modeling approach for this data set, and we pulled out blues for seed weight, which was reasonably correlated across all these environments. So the blues for seed weight were plugged into our FastStack pipeline, and I want to cut to some of the results to explain how this is working. So here on your screen, you can see in grey are the mini-core accessions. So they're GBV for seed weight, and you can see the diversity we have in, or seed size or seed weight in the population. If we jump over to the pink dots, these are the parents that the AI algorithm has picked out that maximize our desirable haplotypes for seed weight across the genome. And so it's really interesting to see, yes, the algorithm has selected some of the best accessions in the mini-core collection at the top there. However, it's actually picked out accessions that have a relatively low GBV, but hidden in the genome are useful or very desirable haplotypes for seed weight that, when reshuffled with haplotypes from these other parents, can create these green dots. And these are the progeny from making the best crosses among these pink parental lines in a double cross F6 breeding program. And mind you, to create these green dots, it's only 2 rounds of crossing. It's about 18 months in practice. And so if you were to do a few more rounds of crossing, you can actually push these green dots completely off the chart, which is really exciting. And so we did this in practice. We actually made these crosses, which was really challenging. Crossing mungbean for the first time. You can see our first cross of mungbean there ever on the left. We're very proud of that. And then we progressed them through our speed breeding pipeline. There you can see a nice picture of one of the plants in the populations. Really nice long pods. And these lines have now been subjected to a seed increase, and we're about to distribute seed of the very best lines based on their GBVs to all of the collaborators in the project, so they can use them as donor material in their breeding programs. So it's which is really exciting, and hopefully these materials will be useful. So coming back to this figure, I hope you can see, there are a number of points in the breeding pipelines that machine learning or AI can be very useful. And this is a huge research area, and there's a lot of opportunity to further develop these technologies as well. We certainly aren't there yet. And so these predictive breeding technologies are rapidly evolving, as you can see. And this really calls for action to make sure that we're training the next generation of breeders with the right skill sets to be able to implement these types of predictive breeding approaches. And so one of the initiatives we're leading here at UQ with all of our partners is a new training center. It's called the ARC Training Center in Predictive Breeding, where we have 5 Australian universities, 2 overseas universities, as well as more than 30 industry and government partners coming together to help train a cohort of PhD and postdoc students, postdocs in predictive breeding. So yeah, it's one of the unique features of this centre is that it is very diverse. So you can see there we have more than 22 species important for agriculture in Australia being worked on. We've got a range of breeding programs or companies involved, from small to medium to large and international companies. And one of the things we're trying to do in this centre is train our students really closely with industry. So all the data that the students are working on is very much from the breeding programs and embedded and part of the breeding operations. So this will hopefully make the research outcomes more applicable for that breeding program, but also the students will spend time in those breeding programs so they get a broader experience and immersion of in the industry environment. And that's something that our partners have identified as a weakness of many of the graduates being produced. Not having that industry exposure has been a bit of a gap that we want to fill. So yeah, this is quite exciting. We now have the centre operational for 12 months, and so we've been busy recruiting a lot of our students. And And one of the things I want to highlight here is our partnership with SRA through the training centre. And we have 3 projects on your screen. And it's actually, all these projects are actually using or connecting to components I talked to in my seminar. So Chris Tom is applying genomic tools for novel nematode resistance in sugarcane, trying to bring in exotic haplotypes for nematode resistance into elite sugarcane. And that's where AI for parent selection could be useful. And Andy Rigby is also looking at genomic prediction of specific combining ability. So can we predict the best combiners that could be the crosses that we make in the future rather than selection based on line performance? And Sujesh is an industry fellow where he's combining UAV captured phenotypes and genomic prediction to see if we can improve the efficiency of genomic prediction, which is quite exciting as well. So yeah, I think that really shows the commitment from SRA for building capacity for the future for the sugarcane industry to be adopting these latest technologies, which is really exciting to be on that journey together. And I guess in summary from my seminar today, I think it's really important to be advancing our predictive breeding technologies. And like I said, there's a lot of opportunity and challenges to do that. But in parallel, we need to be training the next generation of breeders with the right skill sets to implement them is really critical. So I'd like to thank the Hayes Group, my team, our training centres, GRDC, who funds a lot of our grains research here in Australia, and of course all of our partners and funding bodies associated with the 3 case studies I presented today. Thank you very much for SRA for having me on this seminar, and I apologise for going missing for a few minutes there when my teams decided to shut down. Thank you.
Thanks, Lee. That was really a great webinar. I think everyone's agreed. And I think there will be a lot of questions. So if you do have a question, please use the Q&A box, and or if you want to ask a question directly, which is really good, raise your hand and then we will unmute you.
Yeah. My question is, Dr. Lee, how successful has genomic prediction been in predicting specific combining ability when it comes to the non-additive traits, genetics?
Yeah, I mean, I think for a lot of our crops, it's not standard practice to do this, and I think it's an exciting research area to see how we might be able to achieve this, and how does it compare to traditional approaches. So and that's where simulation can be quite useful to evaluate those different approaches in silico before we implement them. So yeah, I think it's something we're exploring in terms of methodology.
Okay.
So yeah.
All right. Thank you very much indeed. Thanks. I really enjoyed your talk. Thanks a lot. Appreciate it.
Thank you. I'll just ask, how's the FastStack impact the genetic variation over time? Sorry.
Yeah, that's a good question. And what we see is when we use the genetic algorithm for parent selection, that the diversity, the global diversity that's sampled in the parental set is much, much higher than parents selected through truncation selection. Often truncation selection results in parents that are really closely related, whereas you're sampling a breadth of diversity for parents selected using the genetic algorithm. However, if you were to do this for repeated cycles of selection, your rate of genetic gain is very, very slow if you're just using the genetic algorithm. So what we see is advantages in actually combining these strategies. So where we might select founders of a breeding population or pre-breeding program using the genetic algorithm, because it's really useful for sucking in all the desirable haplotypes and then applying truncation genomic selection thereafter to start pyramiding them really quickly. And so we find that that outperforms only truncation selection from the very beginning. So that just reiterates, the importance of trying to combine these approaches and tools. And it's not just one or the other. It just really depends on your breeding objective and how can we creatively combine these tools and technologies.
How long do you think it will take for these models to move beyond academic research and be widely adopted by commercial breeding programs? And what do you think is the biggest obstacle to achieving that? Can you introduce an article that compared the power of traditional selection with these new selection methods over several generations?
Yeah. So I think we can't underestimate what large commercial breeding companies are implementing. So multinational companies like Bayer and Corteva have a lot of capacity in this space and are running very sophisticated models and technologies for predictive breeding. I know some of them are already implementing the FastStack pipeline, for example, commercially. So I think that's happening already. There's we're ourselves in terms of research trying to validate some of these approaches. I think with Eric Dinglerson, our colleague here at UQ, has got some really nice validation data on barley, multiple disease resistance. We've been working with a commercial wheat breeding company too for long-reach plant breeders for about 7 or 8 years using some of these tools as well, which is leading to new varieties and impact with those partners. So yeah, I think certainly these tools are working. It's an opportunity to make them even more effective and efficient.
Thank you, Lee. Will it work on adverse conditions like more dry areas such as in Bangladesh, India, this area?
Yeah, I think these approaches can be adapted to any conditions, but it's a matter of what's the crop, the traits, etc. That need to be considered. So it's certainly possible. I mean, we've got new projects even adapting these tools to crops like with our partners with crops like strawberries and even macadamia. I think it's very species agnostic, these tools. It's a matter of adapting, the approach to the species, and every species has its challenge or perk to overcome, and there'll be different traits and things to consider. But I think that's also part of the fun and adventure for plant breeders, with these tools. So yeah.
There is a question from Gary. You mentioned that setting the direction of the breeding program is critical. Do you have capability in your center for plant and crop physiologists to identify optimal design a crop?
Yeah, it's a good question from Gary. I think the biological knowledge or the physiology is really critical, and you can certainly point predictive models slightly off course and maybe end up in the wrong spot after several breeding cycles. So I think that's where approaches that are starting to integrate crop growth models with genomic prediction are very promising, and we've seen commercial success with that approach in the US by Pioneer, now Corteva, releasing product lines such as Aquamax maize, which was led by Professor Mark Cooper. So I believe he's quite motivated to try to develop that technology and adapt it to different crops, including here in Australia. So yeah, I think that's an exciting research front.
Kevin Jacobs asks, what is the standard deviation between the prediction models aligned with the accuracy of the predicted outcome?
Good question. I've got no idea off the top of my head. We'll have to. I'm assuming he's talking about the sugarcane predictions, and if so, then they'll be in the paper. So I have to check it out.
Given how little is known about plant genomes, How can you expect an AI-based prediction to be correct without knowing all the details?
Yeah, well, I think, I mean, to play devil's advocate, plant breeders have made significant advances for our crops, not knowing anything about the genome traditionally through traditional plant breeding. The fact that we now have genomics and the technology is efficient, then it's very useful, and it adds insight around, which haplotypes or QTLs we can combine. But we don't really need to know too much in terms of the underlying genes to exploit a technology like genomic selection. These SNP markers are just markers throughout the genome that are in LD with causal genes or mutations. And so estimating their effects and using a prediction, we know across crops works very well. So yeah, I mean, I think understanding the genome and gene function is very useful and can open the door for approaches like, gene editing or transgenics, etc. And for some traits, that'll be very useful. But for others, more quantitative traits, it's difficult to deploy those types of technologies. Yeah.
I've got a question from Mohammed Usama, which is not the same one, but to what extent do GMOs regulation in certain regions hinder progress in plant breeding?
I'm not sure. I'm not sure if it's hindering progress that much. I think if we look forward, I think it's really important that we have all the tools in the shed when it comes to crop improvement for the future. And so for a technology that's deemed, safe by a global scientific community, I don't see that, there's no need for us to be not adopting those tools and for them not to be acceptable. Why not have them at our fingertips? Because I think that technology is rapidly evolving, and what we can do today is different to what we can do tomorrow, and how we integrate that with quantitative breeding is the real opportunity, I think. That's just my opinion.
Highly appreciated and informative presentation. So this is Dr. Seda Bibi. When do you work in dry lab or machine learning sometimes results in field totally different? How can you control these issues?
Yeah, I mean, I think analyzing data on a computer is one thing, and that's why plant breeders can't lose track of what's going on in the field. They've still got to be in touch with their germplasm, visit field sites, know what's going on in their germplasm to better interpret the data actually on the computer. That's actually really critical. Yeah, I 100% agree.
Charobase, sir, why do we use linear equations of genomic selection when the biological system are non-linear?
Yeah, I mean, it's a good question. I think, there's all sorts of modeling approaches that can be done and explored, and I think that's an exciting opportunity. So, I think the more complex integration of crop growth models and prediction is sort of on that path, and so presents opportunities to tailor predictions to specific environment types for more, better outcomes for the breeding program. So, yeah, no, I think it's a good point and an area for research. Yeah.
Momita, is there any possibility that someday AI would replace the plant breeders in selection of good parents for crossing?
I think plant breeders are safe at the moment. They won't get replaced by the Terminator on my second slide. I think it's a very powerful tool for plant breeders to be using, but it also depends on the scale and size of a breeding program. So a small breeding program, has less resources to be, using all the technologies. And so larger breeding companies have big teams and where they're exploiting big data and making predictions. And they will have data scientists who are running those predictions, as well as breeders on the ground. And so they work together. I don't think it doesn't replace the plant breeder. It's adding value a lot to breeding programs.
Given that biological systems are fundamentally nonlinear and adapted, why do both traditional genomic selection and modern AI systems still rely heavily on regression-based framework? And how can artificial intelligence be integrated with genomics to move beyond the limitation of linear prediction toward biologically realistic, interpretable models of genotype by environment interaction?
Yeah, well, I think, you've got to be sort of strategic and smart around when you use machine learning. For example, we're using a pretty traditional genomic prediction framework in most cases, and we're using machine learning for key steps along the way. There's no reason why these things cannot be integrated end to end. I know breeding companies that have their whole breeding pipeline optimized through machine learning, and it's dynamic. So I think there's certainly opportunities there to connect things. But you need to be very careful and where and how you're applying these approaches as well, so don't go off course.
Yeah, actually, I would like to ask, like, while you developing the prediction model, how is your distribution in the training and testing set? How the distribution you keep to get the accuracy level?
Yep. So I think so it depends whether, I guess, you're trying to build a genomic selection pipeline for the first time, or you're operating an ongoing genomic prediction pipeline. There's ways to optimize your training data set for your prediction by subsetting your data, because you don't necessarily need to use all your data for the prediction. It's about getting as high a prediction accuracy as possible for that subset or those materials. So when you start to build these data sets and layer these data sets in a breeding program, it becomes very useful to exploit. I think I could, there's a lot to talk about there, but I won't go into too much detail. To elaborate, the traditional approach to testing the prediction accuracy is to take a proportion of your population that you already have phenotyped and genotyped and test that prediction by dropping in and out subsets that are included in the training data and not included in the set that you're trying to predict.
Okay, I'll just ask another question and then we'll sort this out. Prediction accuracy is usually higher with the population, however, decrease across the populations. While a plant breeder works on multiple population, what is your opinion on using the prediction models across different populations belonging to different gene pools?
Yeah, I think that's a real challenge, but also opportunity to sort of adapt these approaches. And there's different strategies you might take to do that. So, for example, we know genomic prediction generally works better for predicting germplasm that's more closely related to the training data set. Again, subsetting and pulling out the right training data to inform that prediction or make that prediction on that specific germplasm would be part of the strategy. Yeah.
How appropriate is the use of synthetic data to overcome data scarcity when training ML models for plant genomics prediction, and what approaches can be used to achieve this?
Yeah, that's an interesting question. I know, synthetic data could be quite useful to explore different questions, and So I know, yeah, Owen Powell and Mark Cooper are working with some simulated data sets to investigate those sorts of questions. So yeah, I mean, I haven't had any experience myself, but I think it can be quite useful. Yeah, but maybe get in touch with those guys if you're interested to know more.
Claire,
I'll have to, maybe one more question, Nat, and I'll have to run.
Yeah. Yeah. Sorry, I'll just get this last one from Claire. Have heard AA explained as confidently incorrect. Have you found incorrect outputs in this context?
Incorrect outputs in terms of?
In this context.
In terms of this simulated data, or? I'm not sure.
I've heard AA explained as confidently incorrect. Have you found incorrect outputs in this context?
If it comes to simulated data, I don't have any experience in working with that, and my colleagues do a lot of that work, like Mark Cooper. So you could reach out to him if you're interested. Yeah. And I see a question there about fruit trees, and well, I hope it's useful for sure. I think one of the challenges with any kind of tree is, the generation time, and trying to come up with innovative solutions that effectively, speed breed these species would be a real breakthrough. And so then you can start to combine these tools and use them to recombine haplotypes really quickly. So yeah, we've got projects in the training center on macadamia, which is really I'm really looking forward to. But yeah, I think these tools are really promising for fruit, for any kind of species that has a long generation time. You want to get the crosses right, and selecting those parents is a critical step, because you're going to wait so long to start the next breeding cycle. So perhaps even more important.
Indeed. Well, I know there is like hundreds of questions, but Lee's already giving us a lot of time, and I just really wanted to thank you, Lee, for your really great presentation and for your time. I'll thank everyone for attending this webinar as well. We're very grateful. And, please come to the event page at SRA. You will see in the next webinar advertised. This webinar will be online as soon as we can. Thank you again, Lee. And have a great weekend. It seems like the sunshine is out. Thank you, everyone, and have a good weekend.
Thanks a lot, Nat. Thanks for sharing. Thank you.